hope everyone had a great break. And I would like to welcome all of you who are joining or rejoining us today for a front row seat at the 2021 IWMF Virtual Ed Forum. My name is Sharon Rivet, and I am a WM patient and a volunteer for the Ed Forum Planning Committee. When I was diagnosed in 2018, not only couldn't I say the name of our cancer, but truth be told, I still kind of struggle with it. But I had also never heard of WM. Fortunately, my local chapter of the American Cancer Society guided me to this community, and I cannot express enough what an amazing support and education this group has been to me and my family, and I thank you from the bottom of my heart. It is now my pleasure to introduce Dr. Jorge Castillo from the Dana-Farber Cancer Institute. Dr. Castillo is the clinical director of the Bing Center for Waldenstrom's. He is an accomplished specialist, and he's well known to the IWMF community, and I'm really pleased to say he's also my doctor. For more on Dr. Castillo's bio, you can go to the IWMF website and refer to resources. So today, Dr. Castillo's topic is novel treatments on the horizon and the importance of clinical trials for drug development. At the conclusion of the lecture, there will be a Q&A session. And Dr. Castillo, if you are ready, the floor is yours.
Thank you very much, Sharon. And thank you for the kind introduction. And it's certainly my, honor and my privilege, to help many patients out there, including you. As most of you do know, I'm the clinical director of the Waldenstrom's program at the Dana-Farber Cancer Institute. We're probably one of the largest programs in the country for Waldenstrom's, and we are actually growing as well. We actually have now in the clinic 2 full-time physicians and a full-time nurse practitioner, and we are being helped to run clinical trials by 3 clinical research coordinators and project managers. So we're a bigger family than what we were just a few years ago. And I think that really speaks towards the interest on running specific studies and understanding the biology of the disease, and then transforming or translating this information, into how we can take care of patients a little bit better. For that purpose, I was asked to talk today about new therapies for patients with Waldenstrom's. I'm going to talk about the new stuff, and obviously what the importance is of these clinical trials and these medications, specifically for clinical trial in drug development. So I think for the next 40 minutes or so, I'll talk to you about all that, and at the end I will let open questions, and I'll be happy to address some of the questions. And I think some questions have already been collated, and I'll be happy to address those, and hopefully we'll have a more time afterwards, and I'll be happy to answer any questions as well. So let me share my screen so you can see what I see here. Great. So this is the presentation for today. I'm going to talk about novel therapies for patients with Waldenstrom's, and you can see my email address right there. If you're a patient of mine, you know that you can email me at any time with any questions you have. Your doctors can email me at any time with any questions they have. If you're not my patient yet, you can ask your doctors to reach out to me if you have any specific questions about your case. But unfortunately, I will not be able to provide specific recommendations for patients that I have not seen formally in my clinic. But I will be available to your specialist if they feel that they would like to have an opinion from me at any time. So with that, I'm going to start just talking about clinical trials and why are these studies so important. Clinical trials have been regarded as being the pinnacle of medical research. We know a lot, we learn a lot from laboratory studies. That's where the engine producing science is. And we have to translate what happens to in these laboratory studies into humans. And I think that is the challenging part of the whole process. because when we think about all the laboratory research ongoing in terms of medication development, is like the bottom of the iceberg. It's a very large group of, a list of different agents that are being developed and used in laboratory research. I'm talking about in the tube or in vitro, and then into animals, and sometimes that will be 1-2 different species of animals. And then comes the trials in patients. And that's the component in which, that big bottom, of this iceberg starts getting smaller and smaller to get to the tip of the iceberg, which is the medications that actually are shown to be safe, that are shown to be effective, that are shown maybe to be even more effective than the standard of care currently used for a specific condition. And that's how we advance the medical research. These are studies that are designed in a prospective manner. So we decide what type of data we need to include. And in some cases, like what we do at Dana-Farber, not only the patients are getting the medications and we're seeing how they're doing with these medications, but also, we collect samples from these patients, and then we feed back into the laboratory so we can continue, this, feedback process from the lab into the clinic, into the lab, into the clinic. And I think that model has worked very well, at Dana-Farber, in where I work at the being center from the clinic into Dr. Trion's laboratory and back into the clinic. An important aspect of being on clinical trials is that patients are participants and physicians become investigators. So that is just a little bit different than taking care of a patient, in routine clinical care. Actually, most of the activities are routine clinical care, but there are some activities that are specific for the clinical trial. So we are able to either assess the safety of the patient. We need to be able to test the efficacy of the treatments. So as a participant, then there is just a little bit different, rules of the game, in which you cannot make, for example, changes to your dosing any way you would like to. You have to talk to us first. We need to agree. We need to, support. We need to understand, discuss. You cannot decrease the dose of your medications or change how you take it. Or if you have any specific symptom, even if it's a minor symptom, you need to talk to the doctors, and we will need to, register those, grade them. We have a specific grading system for the severity of the different different symptoms. If you have any surgery, any new medication added, any minor thing change in your regimen, you need to let us know. So we need to have all that information. Participants also tend to be tested a little bit more often than regular patients. And that means more laboratory tests, more bone marrow biopsies, more CAT scans. And so all this you need to know all this very early on going into a clinical trial. So some individuals might not like that and say, I'd rather be a patient, I don't want to be a participant, and for that reason, I don't want to be in a clinical trial. But on the other hand, some individuals might say, that actually sounds good. So how do they know if something is working for me? How do they know really? How do they know if something is attacking me in some way, or could be threatening or damaging to me in some way? And that's how we, move on in that specific sense. Now, physicians becoming investigators is also a different game. We're available, we're happy to answer questions, but the procedures that we follow are really following a specific protocol. So it's not like, somebody comes with a specific finding, let's say a low white blood cell count. The protocol says that we need to do something specifically, right? So every patient with that specific problem will be treated in the same manner. So it's reproducible, the doctors can understand what we're trying to do. So that's how investigators do work a little bit different than physicians. Physicians, we always have the social aspect comes into play when we make clinical decisions, and the medical aspects as well. So with investigators, the protocol aspects have to be there in place too, and we need to abide to protocols and follow the protocols the best way possible. Now, I think a final message is every current standard of care was a clinical trial before. Every treatment that you can see FDA approved or approved by the NCCN or by any other organization in your respective countries, most commonly than not was a clinical trial previously with unknown safety, with unknown efficacy, and participation in clinical trials is what made that unknown, compound into an FDA, NCCN approved medication that benefits, patients all over the world. So from a patient to, benefiting the entire world. That's the idea of a clinical trial. There are different phases, and this is just a very brief primer on what clinical trials are all about. So phase one, this is all in humans. Phase 0 or before, those are laboratory and animal studies, but phase 1 studies are safety studies in which we don't really know if the medication is safe to begin with. So the phase 1 studies are almost always dedicated at analyzing the safety of these agents. So typically we start with lower doses, and then we increase the dose over time, either in the same patient or in a different group of patients. And then we actually are seeing which type of side effects are being seen. And obviously, we do have specific stopping rules if we see that these medications are too toxic. And in that scenario, a lot of medications that in theory, or at least in the laboratory, should work very well, in patients are not safe, even at the minor doses that we potentially could give. And that is a big weed out of potential agents that are effective, but they are not safe to begin with. And that really lead us to understand, how it's different in animals than in humans, because, a mouse might not tell you if they have suicidal tendencies, right? So that is very difficult to assess sometimes in an animal product, what all the symptoms that possibly can occur in humans. Also, the distribution of the medication, the volumes in the levels we cannot translate them one-to-one from animals to humans. So a lot of things go first of understanding if a medication is safe to be given. Once we prove a medication is safe, and then we decide that is a specific dosing that is not too low and not too high to be toxic, then we get into the efficacy part of things. Phase 2 studies, which is probably the majority of studies in wild mushrooms are phase 2 studies, are studies in which we want to look at the efficacy. So what we do there is we say, what is an efficacy that I think is reasonable on this group of patients? For example, I think these patients on any medication today have a 60% major response rate. So I'm going to test if this new medication has an 80% efficacy rate, for example. And based on those are assumptions that we make. We actually say, well, we can enroll 30 patients, 40 patients, 50 patients, 100 patients to try to prove that efficacy. And that efficacy could be overall response rate, major response rate, VGPRs, complete response rate, or progression-free survival. And that's how we basically move on into phase 2 studies. Phase 2 studies are almost always one arm, and all the patients get the treatments. There's no placebos in that specific group of clinical trials. But those are larger studies for medications that have already proven to be safe. And then if the medication is really efficacious, then we can actually go into a phase 3 study. A phase 3 study is a much larger study. And there have been a couple of them, run recently in wild mushrooms, right? The Innovate study and the Aspen study. Those are comparative efficacy, which means that we have a regimen that we consider to be the standard. And then we have another medication that we consider to be the investigational agent. For the Aspen study, the standard was Ibrutinib, and the comparative, the alternative option was Zanubrutinib. And then we basically have a group of patients, and we randomize these patients into each or either group in a way that we have to balance that one group and the other group are actually similar in terms of age, sex, IgM levels, bone marrow involvements, and all the different factors. The reason we do this randomization is to guarantee that if we see any difference in how these medications work, it's because of the medications and not because of something else. It wouldn't be fair if we include, let's say, only patients over 70 in one group and patients younger than 70 in the other group. That will not be a balanced scenario, or only women in one group or only men in the other group. That will not be a balanced scenario. So we need to balance the groups to make sure that hopefully the medication is the only factor that might be playing a difference on these studies. The FDA looks a lot into phase 3 studies, but also in rare conditions, they look into phase 2 studies for approvals. Once a medication passes a phase 3 study, sometimes there's a placebo involved in these treatments. This is when the medication gets approved. Usually, it starts being used in clinical care routinely. And then there's something called phase 4 studies, in which the medication is already out, it's already in the marketing scenario, and then maybe we see unexpected side effects, or is it cost effective, or things of that nature that could potentially affect the marketing or the utilization of these agents. So that's a phase 4 study. Those typically are more population-based, economic-based, and from what. But there are 4 different types of clinical research that most of these medications go through. So with that in mind, let's start talking about ongoing studies in patients with Waldenström's. I think one of the important ones is specifically randomized studies. As I'm telling you, as I said earlier, randomized studies are very important because they actually comparing different approaches. So this is a study that is ongoing in the UK, and I think this study is really something that is going to give us a lot of understanding in terms of where we are currently. So this study is going to combine Ibrutinib and Rituximab, so it's a BTK inhibitor with Rituximab, versus a chemotherapy regimen with Rituximab. So this is going to be cyclophosphamide, Rituximab dexamethasone, CDR, DRC, or any other regimen you want to call it. So this is actually the first study that is going to compare chemotherapy versus a BTK inhibitor in this setting. There's no other study in Waldenström's that have been looked at this. This is the Rainbow study. So 148 patients, previously untreated, are going to be randomized 1-to-1. So that means there's going to be probably 74 patients in each arm. In one arm, the comparator arm, as I said earlier, is the CDR regimen, cyclophosphamide, and the experimental arm is Ibrutinib R. What they're going to be looking at is response at 24 weeks, so within 6 months, to see how the depth of the response is in these 2 regimens. And afterwards, they're going to look at progression-free survival at 2 years. And there's going to be secondary outcomes in terms of the safety, tolerability, overall response rates, impact to next treatment, and quality of life. Quality of life aspects, I think, is very important in every clinical trial in patients with Waldenström's, should include a quality of life assessment at some point. So very excited about this study. You can see in all my presentations, there's a clinicaltrials.gov ID. That's the ID number. You can actually go into clinicaltrials.gov in that website and enter that specific ID, and that will pop up, and it will give you more information if you were interested in participating in studies of this nature. And again, this study is being run in multiple centers in the United Kingdom. Another study that we are very interested is a study with a medication called Venetoclax. Venetoclax is a new agent. When we think about treatment for patients with Waldenström's, and probably you saw that in the prior presentation, we think about chemotherapy, we think about Rituximab, we think about proteasome inhibitors and BTK inhibitors. Venetoclax is a BCL2 inhibitor. I'm going to talk about a little bit more in what that means a little bit later. So this is a new class of medications for patients with Waldenström's. This was a clinical trial that we started in 2015. It was a multi-center study that we did along with Cornell, Stanford, and City of Hope. Patients started at 200 milligrams of Venetoclax for a week, then 400 for a week, and then 800 milligrams for 2 years. These are pills. And then the treatment will be to be stopped. So this is also pills, a different mechanism of action than BTK inhibitors. It's a BCL2 inhibitors, with the idea that we can provide a fixed duration regimen. And that was the idea of the study. So this study was recently accepted for publication at the Journal of Clinical Oncology and should be coming out very shortly. But we do see, and this is all previously treated patients, actually 50% of these patients were previously exposed to BTK inhibitors. So we did see an overall response rate of 83%, meaning 20% of our patients did not respond, but about 80% of patients did respond. And not only that, the majority had at least a partial response, as you can see, the orange and the gray. And the gray is the VGPRs, which is about 20%, 19% VGPR rate with this regimen. So very interesting, very active. And that was the idea of the study. We wanted to show that the efficacy rate was at least 70%. So in this study, 83% was beyond that 70%. So this is a positive study. What we see in the progression-free survival is that about 2.5 years into treatments, at least 50% of the patients have progressed. And as you can see, some of these patients do progress after stopping treatment at the 2-year mark. There's a group of patients who continue, in good response beyond that time frame. So that gives us an idea that we can use Venetoclax in patients with Waldenström's, but I think developing combinations with these agents, with other agents, might be a way to go to make this progression-free survival just a touch longer. But anyway, very interesting, and I foresee this medication being added to the armamentarium of therapies for Waldenström's in the near future. Now, I wanted to talk to you about BCL2. BCL2 is a protein that kind of modulates a specific phenomenon called apoptosis, as you can see here on the right side, apoptosis. Apo means nearby, close by, and ptosis means falling. It refers to in a tree when the leaves get dry and then kind of falls off and it falls nearby the tree. So that's what apoptosis means. And it actually relates to the normal, natural death of cells. So every cell in our body has a lifespan. And these cells, at the end of that lifespan, they basically undergo apoptosis. They basically fade away and they fragment itself, and our body gets rid of it, which is different than necrosis, which we can see on the left side here, in which there is inflammation, there's swelling, there's cell destruction. So that's a different process. That's trauma, infections, burns, all that can cause necrosis. Apoptosis is just a natural process of death. So what happens with BCL2, if a cell has too much BCL2, then they don't die when they are supposed to die. And these cells immortalize themselves. So that's a way in which these malignant cells survive. Actually, BCL2 is increased in over 75% of cancers, human cancers. So it's an interesting pathway to block in the first place. Now, as you can see here, if BCL2 is in the malignant cell, in that protein, the cancer cell survives indefinitely. So what Venetoclax does is Venetoclax basically puts itself in the BCL2 binding site, and by doing that, then BCL2 gets dislodged, and in that situation is free. And when it's free, it can actually induce the death of these cells, and then the cancer cells actually do start dying. So basically, is like very much like chemotherapy is doing in terms of killing the malignant cells. But you're doing this with a pill that is not a chemotherapy agent. So based on the results of the prior study, we have designed this study, which is ongoing. This is an Ibrutinib and Venetoclax combination. These are all oral agents. Ibrutinib is 420, as you can see here, and Venetoclax is up to 400 milligrams. We went a little lower on the dose, but the idea is that if we can treat patients for just 2 years and then stop therapy at the end of those 2 years would be ideal. We're going to enroll 50 patients, and we want to prove that we can get to a very good response rate of at least 40%, if not higher, and with other secondary outcomes. And this is the ID for the clinicaltrials.gov website. So we are up to 42 enrolled patients currently. And there's not much I can tell you about this study, without disclosing data that I shouldn't. But next year, I think we'll be hearing about this study, in a more public manner. So this is an ongoing study, and we're about to complete accrual. If you feel that you want to participate in a study of this nature, please feel free to just shoot me a quick email, and then we can have a conversation. Now, we are now combining Ibrutinib with pretty much anything you can imagine. So Ibrutinib is approved to be used in walnut shrooms alone or in combination with Rituximab. So this is a German study in which Ibrutinib is being added to bortezomib and rituximab. So this is a subcutaneous bortezomib, subcutaneous rituximab, and Ibrutinib. As you can see here, Ibrutinib is given on a daily basis at a standard dose of 420 milligrams. Bortezomib is given once a week, 3 weeks on, 1 week off for 6 cycles. And rituximab is given on day 1 of each bortezomib dose for the first 6 months, which is the 6 cycles, but then continue with maintenance once a month for 24 months. Actually, every 2 months for 24 months. So 53 participants will be enrolled in this study. And if you're in Germany, then you can participate in this study. If you're not in Germany, you could travel to Germany if you want to. There's another study combining a medication called Carboxymab, which is a second-generation proteasome inhibitor, also with Ibrutinib. This is actually a randomized phase 2 study, which sometimes we don't see too many of those, but there are some of them, and this is a good example of that. So this is Germany, again, 184 patients. They're going to be either treatment naive and also relapse refractory, randomized at 1:1. And as you can see here, Ibrutinib is going to be given for at least 2 years. And then Carboxymab is given, 3 out of 4 weeks, once a month for the first 12 months, and then once every month for 12 months. So, and there's going to be a comparative arm, which is going to be Ibrutinib alone. So this is going to be a comparative study, and Ibrutinib will be used as the standard of care, and the combination of Ibrutinib and Carboxymab will be the hypothesis. We have also in the United States a study combining Ibrutinib and Ixazomib. Ixazomib is also a proteasome inhibitor that is taken by mouth, and this is only specifically for relapse patients. The enrollment is suspended currently for interim analysis, so I don't think they are actually accruing patients. And this is the Mayo Clinic is running this with the National Cancer Institute. And again, ibrutinib is up to 24 months, and ixazomib is given 3 out of 4 weeks, throughout. So this is an ongoing study. I don't think it's enrolling patients currently, and hopefully then the interim analysis shows, suggestive data that we could potentially see, next year. We do have another mechanism of action here. Mavurixa 4 is a CXCR4 inhibitor. So we previously had presented some data on an anti-CXCR4 monoclonal antibody, which was given intravenously. The name was Ulocuplumab. Who can keep up with these names? So there's a company that developed a CXCR4 inhibitor, which is a pill form treatment. And this study is specifically directed at patients with CXCR4 mutations. So ibrutinib is given at the standard dose for 2 years, and then indefinitely from then on. And Mavurixa 4 is given at a dose of 200 milligrams by mouth once daily, up to 400. Specific when the safety is, proven, was increased to 400, and now the patients are being enrolled in 600 milligram doses. The idea, at least initially, is to enroll about 24 participants, either treatment naive or relapse refractory. This study is open in multiple parts, multiple centers. I think we have at least 3 centers in the United States, the Dana-Farber and the Anderson in Colorado, and there's also a site in Germany, another site in Greece. So I think this is an interesting study. We were very pleased of how the anti-CXCR4 antibody actually did in the prior study. Unfortunately, the company will not promote the antibody for further commercialization, but then we have this clinical trial using the CXCR4 inhibitor that we're also, keeping an eye on. Some data has been presented already at the ASCO, which is a clinical oncology meeting in the United States, as well as the European meeting, with early evidence of efficacy, but obviously longer term, follow-up is needed to understand if this is a durable benefit, if the responses do deepen over time, which is what we have seen with ibrutinib alone, and if these patients with CXCR4 mutations do well with these regimens. The main focus on CXCR4 is because, as some of you might know, patients with CXCR4 mutations, specifically nonsense CXCR4 mutations, tend to have lower rates of response to ibrutinib, acalabrutinib, and zanubrutinib. There's no data with acalabrutinib. They didn't do any CXCR4 testing on that study. But with ibrutinib rituximab, and zanubrutinib, CXCR4 mutations are associated with lower rates of response. Not a lot of lower rate, but a little bit lower. And we would like to kind of see if we can, get those patients back to, higher rates of response as well with these treatments. Daratumumab is another antibody against CD38. CD38 is a marker that is almost always present in plasma cells. So daratumumab is extremely, effective in myeloma and in amyloidosis as well. Daratumumab now is initially was tried as a single agent in relapse patients, and that alone, in myeloma, and that alone gave about a 40% response rate. And now daratumumab in combination is actually used in the frontline setting in myeloma, is used in the frontline setting in amyloidosis. It has done very, very well for those 2 other plasma cell diseases. And now this study is being done between Cornell and the Mayo Clinic, ibrutinib and daratumumab in patients with Waldenstrom's. This is a small study, about 24 patients. And the idea is if somebody's ibrutinib naive, they could get into ibrutinib daratumumab, or if somebody's on ibrutinib has reached a plateau, then daratumumab is added to see if that can induce deeper response on these patients. So daratumumab is given the same regimen that is done for myeloma, once a week for the first 8 weeks, and then once every other week for the subsequent 16 weeks, and then once a month for approximately 18 doses, and then daratumumab will be stopped, and ibrutinib will be continued all the way to disease progression or an acceptable toxicity. So this study is actively enrolling as well. Now, we were very excited about Daratumumab, and we actually did a study on Daratumumab in patients with Waldenström's previously treated. The aim of the study was to include 30 patients, and the idea was that we hope to see at least a 70% response rate with this compound. When we were up to 13 patients, we decided to take a look at our data because it was our sensation that we were not achieving the responses that we wanted to. This actually was a study that we did in conjunction with the Mayo Clinic, Sloan Kettering, and the Seattle Cancer Alliance. And within 13 patients, this was our outlook. So we saw patients with a 15% partial response rate and an 8% minor response rate with an overall response rate of 23%. So if the goal is 70%, and we're already at almost half of the patients, and we are an overall response rate of 23%, we felt that we were in the realm of futility. We call it clinical futility, because in that sense, that means that each and every patient that we are going to enroll from now on is supposed to respond. And based on what we have seen previously, it's very unlikely that is going to happen. And for that reason, we decided to stop the study early, because we felt it was clinically futile to continue enrolling patients on an agent that was not as effective as we thought it was going to be. Now, there is some people that benefited, as you can see here, a 23% response rate, and at a year, about 25% of patients had not progressed yet. So there is some benefit there, but it was not as pronounced as we would have liked it to be. Actually, we looked at the CD38 expression and how strong the CD38 expression is in the plasma cells of the patients seemed to be the one kind of giving us an idea of who might respond better to these agents than other patients. But again, that is not a practice that is done routinely in clinical care, so it's hard to know how we can translate this data into that. So again, maybe we put the bar too high, for Daratumumab in Waldenström's. I think the study of Ibrutinib and Daratumumab is reasonable. There's actually laboratory data suggesting that if we combine Ibrutinib and Daratumumab, there's some synergism. So I think the clinical trial is reasonable, and if you would like to participate, the ID for clinicaltrials.gov is there, so you can learn more about this study. The Canadians are actually running a very interesting study. Here, combining bendamustine, rituximab, and acalabrutinib. This is for previously untreated patients, the Brom Study. 59 participants will be enrolled. And here, they're going bold. They're going with bendamustine 6 cycles, plus rituximab, which is the standard of care in Canada and many other countries currently for Waldenström's. And they're going to add acalabrutinib for a year. As you can see here, there's no arrowhead here, which means that the study is going to be done at a year. Acalabrutinib will be stopped at that time, and we'll see if this actually induces responses, deeper responses than what we typically see with bendamustine and rituximab alone. So this is for Canadians. That's the clinical study that's going to be run at the national level. The larger centers in Montreal, Toronto, and Vancouver should have this trial open for participation. So if you feel interested by this study, please learn more and ask your doctors about participating. There's another agent called CLR131. CLR131 is a very different type of medication. It's kind of a radioisotope that is given intravenously in 2 separate doses. This is a study that is going to include many different conditions, different lymphomas, but they're specifically focusing on Waldenström's. It is felt that in myeloma and in Waldenström's, for some reason, these radioisotope seems to be very interesting, or specifically more powerful. So 120 participants are going to be included on this study. I do not know exactly what the number of patients will be for Waldenström's alone, but as you see, it's including patients with myeloma and lymphoma in the brain and Waldenström's as well. This study is currently enrolling in many different centers, all over the United States. And then we do have some data that was presented at ASCO by Dr. Ayla Wari, showing that some of the patients with MYD88 wild-type disease had a bit of progression-free survival, but again, the numbers are very small, around 3 or 5 patients. There were three patients with MYD88 wild-type disease, and then 5 patients that were refractory to more than 2 drugs. And so there was some benefit there in terms of decrease on IgMs, as you can see, in about 75% in the MYD88 wild-type patients. About 70% of patients were refractory to more than 2 drugs. So I think more data is necessary to understand these a little bit better. And the major problems with these agents so far has been cytopenias, neutropenia and anemia and low platelets, as expected with radioisotopes, because these radioisotopes also affect not only the malignant cells, but also affect the microenvironment of the bone marrow. But again, a distinct difference mechanism of action here for whoever might be interested on a study of this nature. We do have a study at Dana-Farber now, specifically for patients with anti-MAG neuropathy. This is a study combining acalabrutinib and rituximab. Acalabrutinib will be given standard at 100 milligrams twice a day, for 2 years for the study, and then indefinitely afterwards. And then 4 weekly doses of rituximab on day 1 and day 17, kind of mimicking Innovate, right? Ibrutinib rituximab, but with acalabrutinib rituximab. Now, the beauty of this study is patients with anti-MAG neuropathy will be included. And this includes patients with Waldenstrom's with anti-MAG, but also IgM-MGUS with anti-MAG neuropathy. So we're happy actually that we're able to run a study of this nature. I think this is going to be one of the largest studies ever in neuropathy. It's not yet enrolling, but it's up in clinicaltrials.gov, and we're about to activate here at Dana-Farber. It's going to be so far at Dana-Farber, Mass General study. Is going to be kind of centered here in Boston. The reason for that is because we need to have a very strict neurology evaluation, and so we're going to be partnering with our colleagues at the Brigham and Women's Hospital to have our patients with neuropathy very well, monitored and evaluated, make sure that we're not having other reasons for neuropathy and things that could potentially obscure the study. So this study has 2 outcomes. One of them is to see if the IgM gets better, which we feel confident that that's going to happen. Acalabrutinib has an 80 to 90% response rate in patients with Waldenstrom's alone. But also we want to see at the 2-year mark, 2 years later, if there's any improvement in the neuropathy as well. So we have 2 primary objectives for this study. So we're very excited about this. And if you are interested in participating, please feel free to learn more in the clinicaltrials.gov or shoot me a quick email, and we'll be happy to talk. Now, what comes next is actually very interesting. This actually triplets. In myeloma, we went from double therapy to triple therapy. And this is what seems to be the next step in patients with Waldenstrom's as well. So this is a study that is going to be done, suspended currently because of some FDA comments they're trying to address probably, but it's already running in clinicaltrials.gov, and at some point will be hopefully available for participation. So this study is going to be a phase 2 study, randomizing patients previously untreated to a comparator arm, which in this case will be ibrutinib rituximab, versus an experimental arm, which is ibrutinib rituximab and venetoclax. So this is going to be probably the first study looking at a triplet, non-chemo triplet in patients with Waldenstrom's and comparing it against a non-chemotherapeutic regimen. Again, they're looking at complete response rates, and I think they went very bold with this because complete responses are not very common in Waldenstrom's. But on the other hand, if we don't get to that depth of response, we cannot talk about possible cure in the future. So I think this is the heart is in the right place for this study. And obviously very excited about this too. A number of different secondary outcomes, including adverse events and time to complete response and overall response rates and other classic outcomes of interest for patients with Waldenstrom's. So I'm hoping the investigators will be able to answer the FDA and their comments the FDA are having, so we can start accruing to this study, which I think is going to be very interesting as well. Good. So now we're going to talk about potential studies that are not yet being run. And when I talk about planned studies, I don't know what the rest of the world is thinking, but I know what I'm thinking. So I'm going to tell you what we're thinking at Dana-Farber in terms of clinical trials for the near future. What happens to patients who have been exposed to ibrutinib, acalabrutinib, or zanubrutinib? Those are called covalent Btk inhibitors. is that if these patients do become resistant to these medications and the IgM starts going back up and we need to switch treatments, most of the times these progressions are driven by BTK mutations, specifically this mutation called C481S. So what happens is this mutation comes into place, and then it doesn't allow these drugs, covalent BTK inhibitors, to bind to their target, and in that way, these cells become resistant. So there's a new group of medications, also BTK inhibitors, but these are called non-covalent BTK inhibitors. Non-covalent only means, what it means is they have a different way of binding to the same target. So instead of binding using that 481 when the mutation occurs, these new BTK inhibitors bind in a different way that can still affect the BTK, but do not need to interact with 481. And for that reason, it's possible that these agents can work even though these patients are already progressing on ibrutinib. So this was a study that was recently published, and as you can see here, a lot of different conditions, but I'm going to bring you to the WM group. And as you can see, the dark blue lines, or the darker blue lines, are the patients who were progressing on a BTK inhibitor, and then they went into pirtobrutinib. This was also called LOXO 305. And as you can see here, 6 out of the 8 patients actually had an improvement in their disease. So this is very intriguing and very interesting. So we are planning a clinical trial with pirtobrutinib in patients with Waldenström's who are either progressing on covalent BTK inhibitors or intolerant to BTK inhibitors. That means if patients are having, grade 3 or 4 side effects or severe grade 2 side effects, then they will be able to move into pirtobrutinib. I'm hoping to have this study open sometime probably in the 3rd quarter of next year, but this is something that we're planning, just to give you a heads up. And now we have gotten to the age of immunotherapy. Loncastuximab is an anti-CD19 antibody drug conjugate that has been approved recently for the treatment of diffuse large B-cell lymphoma, which is a much more aggressive disease. And we already have a couple of patients who had Waldenström's and then progressed or transformed into diffuse large B-cell, and they got treated with multiple treatments. And there are actually some of them, a couple of patients are in loncastuximab doing very well, actually, not only from the diffuse large B-cell, but also for the Waldenström's aspect of things. So this is the structure of the antibody drug conjugate. Here on the left, we have the antibody, and we have this payload, which is here, these yellow hexagons. So this payload is actually inactive when it's bound to the antibody and is floating in the bloodstream. So it's not active when the patients are being infused. These are intravenous treatments. But these antibodies directed against CD19, and CD19 is expressed in all Waldenström patients, in the cells of all the Waldenström patients, the malignant cells. So these cells will basically uptake the antibody and will introduce the antibody into their cytoplasm. They will chew in, they will eat, engulf the antibody. And when the antibody is in the cell, that's where the payload gets released. As you can see here, the payload is called tesirine. It's basically an alkylating agent, and it's microdoses to the point that the tesirine just gets in, the antibody gets released and then degraded, and the payload gets into the nucleus where it causes DNA, cross-linking problems in a way that the malignant cells cannot reproduce and they die. So my colleague, Dr. Shena Saroshek, she's a junior faculty in our clinic. She's going to be leading this immunotherapy study. It's going to be probably the first immunotherapy study in patients with Waldenström's, and we're very excited about this study as well. I'm hoping that this study could be open in the 1st quarter of 2022. So again, if you want more information about this, please feel free to reach out to me as well, or have your doctors reach out to me. I'll be happy to chat more about this. So just some quick conclusions. There are several exciting clinical trials that are currently ongoing. We have BTK inhibitor combinations, doublets, triplets. We have non-covalent BTK inhibitors. We have BCL2 antagonists coming in, anti-CXCR4 agents, and there are other pathways that we haven't talked about yet, HCK in IRAK1, IRAK4. So immunotherapy is up and coming. We haven't talked about something called biclonal or B cell engagers, bispecific T cell engagers, or BiTEs. These are another type of immunotherapy that also is very interesting for patients with Waldenstrom's. I think we're going to see really, the work never stops. So there are many more treatments coming up, and I think all these treatments will translate, hopefully, into better treatments, safer treatments with more durability in patients with Waldenstrom's. So just my couple of comments here: always consider participating in clinical trials. That's the only way to advance the science. But do your homework. Go into the websites, read about it, talk to your doctors, learn more about this. If it makes sense for you, then we're happy to help. And I think your participation is key. Without patients participating in clinical trials, we cannot advance the field in any reasonable way. And with that, I want to thank you for your attention, and I will give way then to the questions. Thank you very much.
Wow, thank you, Dr. Castillo. That was a lot of information, but it was very helpful to provide this view into potential new treatments and just how important participating in the trials are to really propel drug development and look at the research, which is so beneficial to many of us. So thank you again. And if you're ready, we have some questions that were submitted in advance, and I did get a couple through the chat. So if we have a chance to get to those questions as well.
Let's do it.
So our first question is, now that safer drugs are available, is it still beneficial to delay treatment for WM until the symptoms are not manageable?
I would say still the right approach for a patient who is asymptomatic or minimally symptomatic is not to treat for a number of reasons. So if we have an asymptomatic patient, we actually have a study that we followed 400 and some patients over time, and about 80% of patients needed treatment within 10 years of treatment, of diagnosis. But about 20% of those patients did not need treatment for longer than 10 years. So I think, in that setting, to think about, can this patient who is asymptomatic today be one of those 20% who will not treatment for more than 10 years, right? So I think giving that chance to the patient is very reasonable. On the other hand, there was a recent study from the Mayo Clinic that came out in those patients with asymptomatic disease. And when they compare the life expectancy of patients with asymptomatic disease not yet treated, the life expectancy was very compatible, similar to other individuals of the same age, sex, and race, in this country. So if you have a normal life expectancy at that moment, what is the rationale to treat, right? So I still believe that in patients who are asymptomatic or minimally symptomatic, waiting and observing is the right thing to do.
Okay. All right. Thank you. Some of the common drugs used for WM have never been evaluated in randomized clinical trials in WM patients or received the regulatory approval. How is it decided which drugs require randomization clinical trials and regulatory approval specifically for WM? And then second part to that is, which drugs can be used based only on approval for other B-cell neoplasma and clinical experience?
So kind of a long question, but I mean, bottom line is, in patients with Waldenstrom's, the first approval was Ibrutinib. That was not a randomized study. And the reason that was the case is because really, it's so rare to see randomized studies in patients with Waldenstrom's. Actually, up to the moment in which Ibrutinib data was published in 2015, I think there have been only 2 randomized studies that I'm aware of, and none of them really focus exclusively on Waldenstrom's. More recently, have been a couple of randomized studies, right? We have the Innovate study, which is Ibrutinib rituximab versus rituximab, and then the Aspen study, Zanu versus Ibrutinib, and that led to the approval of Ibrutinib rituximab by the FDA and also Zanu by the FDA. So I think my sense is, as we have shown that we can run randomized clinical trials, then I think more randomized clinical trials will be done, and obviously the regulators might require more clinical trials, randomized studies for that purpose. Having said all that, FDA approval is one good thing to have, But in the United States, we also have the National Comprehensive Cancer Network guidelines, which basically include every potential treatment that show benefit in patients with Waldenstrom's. And that includes not only, the FDA-approved medications, but also other agents that have shown, at least in clinical trials, to be effective. So bortezomib is there, bendamustine is there, carfilzomib, ixazomib, acalabrutinib is there too, even though those agents are not formally FDA approved. So I think there are different ways in which we move around this. I think FDA approval is important. I think ACCN endorsement is important too. So as long as there's a clinical trial that has been done, I think it's important to use those medications. Now, in a time in which we didn't have any other options and the disease were too rare and there's no clinical trials, then you can actually start transpolating from other diseases into Waldenstrom. But at least at this time, I don't think we can say that, if this medication is active on this disease, then it's going to be active in patients with Waldenstrom. I don't think we can say that anymore. With chemotherapy, it's much more general, and I think that could be a statement that we have done previously. But with targeted agents, I think they are very specific, very disease specific. So I don't think we can be transpolating these data just blindly. I think it's important to have the clinical trials done.
Thank you. That was a long question.
I know.
All right. Under what circumstances would you recommend that a WM patient participate in a clinical trial?
I mean, there are clinical trials for, as you have seen there, for treatment-naive patients, for patients who have been previously treated. So any patient could participate in a clinical trial at any time, right? Except the patients who are asymptomatic, right? We don't have a clinical trial for asymptomatic patients yet. So I mean, if the patient is interested and the patient feels that the patient will benefit from a clinical trial or is interested in a clinical trial, I think I would recommend a clinical trial in any sense.
Okay. I believe this next question is okay. In order for a patient to participate, is it necessary to exhaust all of the currently available regimens first?
Well, based on what we have seen, not necessarily, right? I mean, even if you are a previously untreated patient, you could potentially be on a clinical trial as well in that specific setting. So I see how in other conditions, exhausting all potential options need to be done first before getting into a clinical trial. But there are many conditions nowadays in which that doesn't have to be the case. If you are in a specific setting, you will find a clinical trial that you could participate in.
Okay, thank you. All right. After a clinical trial ends, are there ongoing communications with the trial participants?
I'm assuming between the investigator and the participants. I think that's what I'm assuming. So the answer is yes. I mean, most patients actually continue coming back to see us after the clinical trial ends. They go back to their primary oncologist, and then we continue being the supervising oncologist for the management of the patients. So yeah, we continue in very close contact. We continue being available to our patients, answering all their questions and answering all their emails and messages. So yeah, we're very much involved with the care of patients after the study is done.
Great. Okay. And next question. Yes, this one was specific to Dana-Farber. Yeah. So to participate in a Dana-Farber trial, is it necessary to physically travel to Boston for each scheduled checkup? Then the next piece of that question is, can it be done through local doctors without travel? And if it is necessary to travel, how often do patients usually need to come?
Yeah, so if the study is only open at Dana-Farber, then yes, it's necessary to come to Boston and be seen here. Typically, cannot be done through local doctors, because there's specific regulations by the FDA and other centers in which, we are the main supervising doctors, then everything has to be done here and be seen here and all that. Now, each trial is different. We do have trials in patients, for example, come once a month for 6 months and then every 3 months after that. Sometimes we have patients who come, the first couple cycles is, weekly or every other week visits. So, I mean, it varies greatly. So each and every study is different. So I cannot tell you there is one specific frequency that applies for each and every study. But yeah, now if the study is open in other places and you come to see me and you have a study that you're interested closer to you, then I typically refer the patients to be seen in other centers that could be more convenient for the patients to go to. But yeah, clinical trials in a specific center, the patients need to be seen there.
Okay. Our next question. Okay. So to participate in your Mavurixa trial, is it necessary to have a CXCR4 mutation? So that's the first part. And then if a patient has had a previous bone marrow biopsy without CXCR4 testing and is now considering your trial, would you recommend another bone marrow biopsy to see if they are eligible?
Yeah, that's a good question. Yeah, so the patient needs to have CXCR4 mutations for that study. And even if the prior biopsy was negative, there's some details on how the CXCR4 testing is done. So our detection rate at Dana-Farber is a little higher than the average because we do have other techniques that we use to find that mutation. So it is possible the patient might have the mutation. It's just that was not identified previously. So if the patient really wants to be on that study, the patient will need to come here. We will need to do a bone marrow biopsy here, and we'll need to run the CXCR4 mutation in our laboratory. And if it's negative in our laboratory, then the patient will not be eligible. But if it is positive, then the patient will be eligible.
Okay, thank you. Okay, do you see a role for CAR T cell therapy for WM, perhaps as a treatment of last resort? And then what are the toxicities of CAR T therapies?
Yeah, I do see a role for CAR T cell therapy at some points. The issue as of now is basically in the question itself. I mean, they are very toxic approaches. And so far for incurable diseases like myeloma and CLL, there's no cure with CAR T cells either. So we're not talking about a curative treatment at this time. And therefore, there's another option for treatment. So we're still trying to choose what the best product is going to be. There are multiple products out there targeting CD19, CD20, BCMA, so many different targets out there. So we're still trying to decide what is the best target for patients with Waldenstrom's in that specific sense. Doesn't have to be a last resort, but probably it will be at the beginning because of the toxicity associated with this. And again, toxicities are varied greatly, but the classic toxicity that we worry about is this condition called CRS, which is the cytokine release syndrome. Cytokine release syndrome is just a systemic inflammatory condition that happens when some patients get exposed to CAR T cells. And for that reason, patients on CAR T cells need to be admitted to the hospital. They need to be monitored for the first several days on these, of these therapies, specifically because they might develop this CRS condition that needs to be treated acutely, and it could potentially be problematic, with brain inflammation and infections and so many other problems. But again, as the technology gets better, right, I think it will be easier to use and maybe easier to manufacture, easier to give. So yeah, at some point I see a role of CAR T cells for Waldenstrom's.
All right, well, I think we have time for one more question. So, come up. So I will just ask, there were several more questions, but I'll just ask, oh, there it is. All right, there is a trial with Daratumumab plus Ibrutinib. Do you foresee a trial with Daratumumab plus Rituximab to target both CD19 plus and CD38 plus cells at the same time?
Yeah, I think that's a very reasonable combination. It's just that it's going to be a lengthy process because the infusions with rituximab takes hours and they are weekly, and the infusions with daratumumab takes also hours, and they are also given weekly. So, I mean, I think, theoretically speaking, that combination, it could potentially be very interesting. It's just that the clinical trial will need to be designed in a specific way, and I think will be, there has to be a lot of time commitment for a study of that nature. But I think it's reasonable. It's a reasonable idea to have.
All right. Well, Dr. Castillo, I want to thank you so much for sharing your knowledge, for answering our questions, and for everything you do for this community. We truly appreciate it. And on behalf of the IWMF, I want to extend gratitude to the EdForm sponsors, Beijing, Pharmacyclics, Janssen, Selectar Biosciences, X4 Pharmaceuticals, and the Treadway Foundation. And thank you to everyone that joined us today who's watching this educational program. We appreciate it. And our next presentation will be starting at, just double check here, 2:30. So please come back and join us for our next session. And again, Dr. Castillo, thank you so much.
Thank you very much to the IWMF for the kind invitation. I really enjoy this type of interactions with patients, and I'm hoping that next time we'll see each other in person as we used to.